Okay, I think we are ready to get started. Good afternoon, everybody. For those that do not know me, I'm Gus Kousoulas, a professor here at the Department of Pathobiological Sciences, and also the principal investigator of the Louisiana Biomedical Research Network. This is the second seminar following the lecture I gave last week as part of the departmental seminar and also the LBRN network on the molecular and cell biology of coronaviruses. That seminar was recorded and it's available on the LBRN website for your view. This seminar also given by Elia Brodsky will also be recorded and will be available at the LBRN website probably within a few days. The coronavirology obviously is of quite importance right now, as evidenced by the fact that a lot of people have already looked at our lecture that was given last week. And during that lecture, we delved into some of the bioinformatics tools that could be used to look at the phylogenetics of the virus as well as the structure and function of different proteins. That was done in collaboration with Pine Biotech, which is a spin-off of the Tauber Bioinformatics Research Center of the University of Haifa in Israel. In fact, a lot of the software and pipelines have been built by the Tauber Center, and they're available through Pine Biotech in collaboration with the Louisiana Biomedical Research Network. We have offered a graduate course in transcriptomics, which is nearing towards the end. Final exam is happening right now on advanced transcriptomics and RNA-seq. And we will be offering a bioinformatics for infectious diseases course that will be available for all undergraduate students through the LBRN network this summer. We are trying to discuss the logistics of this, and we hope that we will have that started sometime in early June. At this point, then I want to transfer the control of the presentation to Elia Brodsky, and you can take it from there, Elia.
Okay, thank you, Gus. And thank you everyone for joining today. What we wanted to do after last session that Dr. Kousoulas did was kind of go into the analytical tools that we are trying to make as accessible to the wider community to study publicly available data as well as data that's generated in different labs. So we'll talk a little bit more about these tools, and I'll provide a few different examples. All of these tools are available on the platform that we use both for education and for research. And the purpose of this platform is to make some of these publicly available tools and custom scripts and custom solutions easier to use for those that don't have an engineering or a computer science background. That's one purpose. The other purpose is to really make those tools work for larger data sets and make it all streamlined in a single location. So in the context of these tools, as Dr. Kousoulas mentioned, we are finishing right now a graduate course on analysis of RNA-seq data or transcriptomic data. This course on bioinformatics for infectious diseases will also touch on a lot of the elements that we'll discuss today, how to deal with different mutations that accumulate in different strains, how to compare them, how to look at protein function and conservation. And so what we did is we offer these free workshops and webinars throughout the different universities in the state, but also we collect all of these resources into this centralized portal, which can be found on lbrn.t-bio.info. So if you go there, that's where this course is. That's where information about this course on bioinformatics for infectious disease will be. And also on that, we are developing a collection of online asynchronous courses that you can use to have undergraduate, graduate students learn about the different data types and different analysis methods And we are working with LBRN members to make those implemented in different types of courses. So some of the analysis methods that we'll touch on today have direct applications to study of virus replication, evolutionary studies about virology, but also the response. And so I'll start, because we just talked about RNA-seq, I'll start with that section that we have on transcriptomic data analysis, which could be used to understand the expression of different genes in response to viral infection. And also we can compare different immune responses. So those types of analyses generate a lot of data for different genes and different pathways that are expressed. And one of the sections on the platform is dedicated to making sense of those interactions that might be fairly complex. And so there's network analysis and machine learning tools to help understand those patterns and identify specific genes and pathways and signaling regulatory networks that help explain those types of responses to viral infection. For example, one of the projects that we will discuss in the context of this Bioinformatics for Infectious Disease program has to do with a study. All of this is, by the way, taken from publicly available data sets. So that study was a comparison of SARS, different defective genomes that have mutations in ORF6, as opposed to wild type, and compared to mock infection. And what we can clearly see here on this image principal component analysis, a difference over time. These samples were taken in passages, I believe 64 passages, and so you can see a very clear separation as the time progresses along the principal component 1 axis. So those types of studies are possible, and I think can reveal a lot of important information about the structure of different proteins and the function, as well as the different responses to cell infection. Now, the platform itself is built to eliminate the need for coding, but also to, as I mentioned before, make the process more streamlined and kind of incorporate some logic into the analysis. So it's not just running tools that you know about, it's also understanding how different tools can be combined together into pipeline, what is the logic behind organizing them in a certain fashion. One of the things that we will also talk about, okay, something here. Okay, so we will talk about analysis of mutation variants, and I'll talk a lot more about this today in a little bit. An important aspect of that is, of course, understanding not just the, for example, presence of different mutations, but also the impact of those mutations at the nucleotide level on amino acid sequences and the change to the protein function and structure. And so in the context of that challenge, we've assembled a number of tools for data association and evolutionary studies that include some simple to use tools for phylogenetic analysis and association between phenotype and changes at the genetic level. So we'll talk about a couple of examples using that section. So for example, one of the obvious questions that could be asked, how is COVID-19 related to other coronaviruses? And here what you can see is a comparison between equal numbers of samples in different groups that reveal that COVID-19 is the most closely related to the SARS coronavirus. Now, as you generate these types of comparisons, it's important to link the information at the nucleotide level, at the whole genome level, to where those mutations accumulate, what are the most pronounced differences, and look from the standpoint of structure and function. And so to help understand and interpret how these things could be studied, we designed a number of visualization tools that we'll also cover. And so what they help do is map these different mutations on structures of proteins, and also to be able to compare, for example, when we talk about vectors and hosts, how can we analyze the differences in those structures and understand those from their evolutionary perspective or from the impact on the function of the proteins. Besides these standard tools, what we would call standard because they apply to genomic and transcriptomic data, so next generation sequencing data, an important part of understanding the different processes involved is being able to model them using mathematical modeling. So the Tauber Bioinformatics Research Center designed a number of tools that can help us translate the data that we have from different experiments, not necessarily based on genomic data, but also based on other types of information to mathematical modeling that could be then simulated, and other tools that are going to be important for that as well. So for example, quantification of viral particles using GFP from microscopy images, and also screening and docking of 3D structures. So the goal of the platform is to really provide a broad overview of the different types of tools, different types of data, and to provide methods on processing and then integration of those together. So for example, I'll just briefly mention some of the preliminary studies that are being conducted at the Tauber Bioinformatics Research Center using some of these tools. Identification of mutations that appear in structural proteins like the spike glycoprotein, as well as the RNA polymerase, to identify how those are different between, for example, SARS and the SARS-2, or between different geographic locations, and what might be the implications for drug screening, vaccine design, and understanding of how that virus mutates as it spreads throughout the world. So some of the collaborations that they have are with University of California, San Francisco, Institut Pasteur, and the Georgetown University School of Medicine. And so one of the things that I think it's important to mention in this context is that with the available grants and research support from universities, a big part of taking advantage of that is having some preliminary data based on maybe publicly available data already, and then combining the resources from different universities to be able to generate new data to inform these kinds of projects and taking them further. So I think it's an opportunity if anyone is interested to discuss research collaborations, not just using the tools, but specifically with a team of bioinformaticians there. I would be happy to connect you with them directly. Okay, so to show how some of these tools are going to be useful to apply to publicly available data on COVID-19 and similar coronaviruses, I've prepared a short little example that we can review in this session to understand how these tools function. So what's something that probably most of us have noticed is that there are a lot of new resources and data sharing platforms that are popping up in different universities to facilitate the discovery of basic questions, answering basic questions about COVID-19 and design of different interventions. So we specifically did a review of genomic resources for virology research and education, which is a blog post that we published. It's mentioned right here, and kind of looked at the rate of publicly available data being submitted on NCBI and other public resources. And we noticed that there's at least a fourfold increase in availability of data, the rate of that data becoming available. And almost all of the prominent universities around the world, one of the things that they're trying to do is to collect, organize, and annotate this publicly available data and make it useful for the different research teams to kind of get started and produce some preliminary results. So I think that in that context, what we have available is a lot of high quality data. And so I briefly wanted to look at one example. On NCBI, there is a new section to NCBI called NCBI virus. And on NCBI virus, it's very easy to find some of those publicly available data sets. One important thing that you probably saw in the news is that there is a collection of samples generated by Oshner, and also those genomes have been made available. So if you go to NCBI virus, you can find different genomes, and here are a couple of them from New Orleans. So I think, making it relevant for all of us as well. What type of information do we have on NCBI about these different viral genomes? They are organized by strain, country where they were collected, different hosts, bats, humans, civets, others, as well as the GenBank session, which allows us to download the whole sequence. And so what we can do using this publicly available data is use a section that I wanted to briefly demonstrate that allows us to compare using multiple sequence alignment those genomes, either full genomes or specific proteins from that repository. And really understand a number of questions about how the fidelity of replication and appearance and accumulation of different consensus mutations or random mutations can help explain the evolutionary relationship between different strains. And so two types of analysis could be done. One based on FASTA files that already contained assembled genomes, and those assembled genomes have a consensus from the sample of multiple viral genomes present in that sample, or directly sampling FASTQ files that contain reads, as shown here, and organizing those reads by haplotypes or by groups of similar genomes that contain minimal variation within the group, and comparing those to some reference genome. So to understand kind of the concept of haplotypes, we can look at, this is, again, just a multiple sequence alignment example, where we can see that the different groups contain more similarity to each other, but they're fairly consistent within a group. So that is an important part of analyzing data that is coming from patients, for example, because if we look at the conservation of COVID-19 genomes overall, there's not that many changes that actually happen in the context of this pandemic. But if we look at the detailed data from these samples from patients at the FASTQ level, at the individual reads, we do see a lot of variation. And that kind of variation can explain, we hope that it can explain the origins, but also the different types of impacts that will have on how these viruses infect patients as they mutate and develop. So an important next step would be to understand how those nucleotide variations affect codons and help us interpret whether those mutations have a significant impact on the protein structure. And so I'll just go through some of these tools and show an example. So I'll just switch over to the platform here. The platform, again, the collaboration with the Louisiana Biomedical Research Network allows you to explore the platform tools and learn about how they work. But also you can get a research license to utilize these for your research projects. And a first step is often to just understand, well, what's inside each section. So we prepared a number of demo pipelines where you can just kind of play around with the tool and understand what it does. So here, the section on virology has been separated. And when you get to the platform, and I'll probably just post this link in the chat here so you can take a look. Here, when you get into the actual platform, you would have to start by uploading your data. But we'll just look at some prepared analysis that actually does not require any upload. So to get there, just click on this drop down, and we can take a look at some of the different sections. So as I mentioned, there are two main sections. One has to do with patient data sets, where you get FASTQ files, and those FASTQ files contain different reads for each sample. Or you can take a collection of FASTA files, so assembled genomes, and those assembled genomes could be compared in some sense. Everything starts, let me actually do, I'll go to the other one. So this is a process of comparing mapping first on the reference genome, right? Because we have FASTQ files and we have reads in them. So the first thing that you would do is mapping, and then identify point mutations, and then organize them to identify haplotypes. In the other section, you would start by loading the files and doing multiple sequence alignment. So as an example, we have here a collection of genomes from different species, and we can do multiple sequence alignment, convert those to amino acid changes, and understand how, as you know, the differences in codon changes have an impact on the amino acid, but also are distributed in different ways throughout the genome. And we have a number of phylogenetic trees that you can build using BEAST or APE, or it's also possible to just run all of the phylogenetic methods and then analyze the differences between the phylogenetic trees to select the one that is most representative of the evolutionary processes that we think are happening. So as a result of this simple pipeline, right, where we did multiple sequence alignment and then built a phylogenetic tree, some of these outputs would look like this. We will have a comprehensive collection of nucleotide changes compared to their codon and amino acid changes. And this is an important output that could later on help us select, for example, specific regions in the genome where we would like to look in further. But from a global perspective, what we would want to do is just take a look at some kind of a visual output that helps us understand what are the differences found in general for all of these different samples that we put in. So here, as I mentioned, as I showed in the previous slide, you know, it's easy to start looking at those distances between groups of samples, right, and arrive at some general conclusions. So what are the conclusions that we see here? Here, I prepared the files in such a way that they were put into different groups according to the different coronaviruses, so beta coronavirus 1, COVID-19, MERS, and SARS. And you can see that the phylogenetic tree shows similarity, right? So evolutionary distance between those different groups, and the COVID-19 is the closest to SARS. Specifically, whether this gives us a lot of useful information at this point, we don't have that. But what is important is to then use this information to start going deeper into why and how are they different, or how are they compare to each other. So a lot of those questions could be answered by looking at the regions where those mutations emerge. So let me just briefly show you a visual output from this pipeline. As I mentioned, we developed a visualization tool that we intend to use for the different types of comparisons that are meaningful. And so here we have some preloaded comparisons. that you can easily navigate visually. And so this is the comparison that we just looked at, right? And so I mentioned that one of the outputs is that output that helps link the nucleotide sequence to the amino acid sequence to then different proteins. So here we have the reference genome, and these are organized into different bins. You can make those smaller or bigger. And the colors show the number of mutations in that bin. So for example, and by the way, here on top, we have the different proteins. So this is the spike glycoprotein, for example. This is the ORF1AB polyprotein, and others. And so what we can see right away is how these different groups that are encoded in different colors compare to the reference genome and compare between themselves. So this red one is the COVID-19 group, right? The reference genome here is the COVID-19, so it is the least, the most similar to its reference genome, of course. But then you can see that the closest is the SARS, and maybe the other ones are the most dissimilar. You also right away see that some of these elements are going to pop out. So here we can see a lot of red. And one of the things that you can easily identify here is that this is just an incomplete genome because a lot of these positions are going to be missing. But what is interesting for our comparison is that a lot of these differences are right here under the surface glycoprotein. So here we can click here and understand a little bit more. What is this protein? What does it do? What is its structure? Kind of look at it. We have links here to the original submission on NCBI. And you can look at the amino acid level, what's going on. So by visually navigating through the sequence, we can start looking at specific regions where those different groups of samples are most different and start asking questions, how are they different? So starting from here, I would like to just briefly demonstrate how these types of comparisons can help reveal some of the more functional implications of these mutations that are accumulating by doing those kinds of comparisons. So here we see a comparison between human isolates of SARS and COVID-19. You can see that the changes are distributed throughout the whole spike glycoprotein that is responsible for the cell entry, and compare that with the bat coronavirus rat G13 genome versus the COVID-19. All of a sudden you see that those mutations are organized in specific domains. And so now it would be interesting to start asking those questions. Well, how are these different proteins functioning, and what are those specific domains? So what you can see here is those same mutations on the spike glycoprotein, but now we have another model that is already bound to the ACE2 receptor. Now it's actually, this is where you see the mutations, but they have 3 different regions, and we'll look at that in a little bit. And now you can see that this is the receptor binding domain, and so those mutations are probably responsible for the differences between the bat and the human ACE2 receptor. And so for the virus to adapt to a new host, obviously those are the important changes. However, that's not the only region, right? We see also some other regions as well. And so some of those things are going to be important. We'll talk a little bit more about some of those other regions and what types of mutations we can find there. But this is a comparison of those FASTA files, right? So those are whole genomes, consensus genomes from different samples. A lot of times we want to look in greater detail. And so as we talk about this pandemic and those types of analyses that are done between all of the COVID-19 samples, we now know that there are multiple clades that could be detected throughout the pandemic. So this is a figure from GISA, where at least 3 specific strains could be identified and linked to marker variants. And so those marker variants could be found here. You can see in different proteins, in ORF8, the spike glycoprotein, and NS3. So how can we use the FASTQ files to identify what types of mutations, right? The question is, how can we take a group of patients, for example, from US and from Australia? This is an analysis that was done at the Tauber Bioinformatics Research Center, and here is the result, where 9 patients, again, this is publicly available data, here are the bioproject references, patients from the US and from Australia are compared to identify the most significantly different mutations in terms of their frequency. How often do they appear in the reads of those samples? And you can see a number of significant mutations, specifically in the spike glycoprotein. So those are interesting because they could help us understand how the changes throughout the pandemic affect cell entry, cell fusion, and potentially the replication process inside the cell. So to briefly speak about that, we did another example here, and I'll show some of those specific mutations that we were able to look at by comparing the different SARS-2, so the novel coronavirus SARS-2, and the original SARS samples from 2004 and 2017. If you look at the genome in total, what you're seeing here is a similarity score between the multiple sequences that are aligned. And here are specific regions with a lot of dissimilarity. So this is a region that is expanded right here. And you can look at, in one of those outputs from the platform that I was referring to earlier, the frequency of those mutations found in specific regions. So here we see the nucleotide sequence, and we can think of this nucleotide sequence having an impact, so being more prevalent in different areas on the spike glycoprotein. So for example, what types of comparisons can we make? This is the spike glycoprotein that consists of the S1 subunit and the S2 subunit. They have different functions. So this is for binding and this is for cell fusion. And so one of the questions that we can ask, well, which one is more mutating, right? Which one has more mutations, just by looking at the different portions of the spike glycoprotein. But just the structure of the protein is not all. We also know that it is glycosylated, and so it's covered with sugar molecules, making it more difficult to be recognized by the immune system, and also that has a role in the binding to the ACE receptor. So that's another area where we can look and try to identify, well, how does the glycosylation change between the different strains? And finally, we can look at very specific regions. For example, in MERS, sialic acid is a pocket that is going to be important, right? So we can look into how very specific pockets change as a result of these mutations. So just briefly kind of give you an overview of the spike glycoprotein. It's located here after around 21,500 nucleotide position. And it has the signal peptide, the receptor binding domain, and those two are in the S1 subunit. And then the S2 subunit has the fusion protein, heptad repeats, transmembrane domain, and the cytoplasm domain. So the main, like I said, separation could be made based on these 2 subunits. The 2 subunits are cleaved in this red site right here. And they're next to the fusion protein, the heptad repeats that are down here in the S2 portion of the protein. So what's an important finding from comparison of these different SARS coronaviruses is that there's an insertion that is happening right here in the cleavage site, right? So one of the questions that we might ask is what kind of an impact can an insertion play in terms of how these two portions of the protein work together, right? And this is a different type of change. It's not a substitution. It is an insertion of a number of amino acids. Another important portion of the S1 subunit is the receptor binding domain. So when this is from the publication where the structure was solved from the novel coronavirus, spike glycoprotein, what you can see here is that in general, the whole S1 subunit is more variable than the bottom S2 subunit. And the most variable region that we expect to change is, of course, the receptor binding domain that actually has a very specific function in binding to this ACE2 receptor, but also it is dynamic, right? It's not a stable condition that it's in. And so here you can see some of the nucleotide changes. There's quite a lot of nucleotide changes in the receptor binding domain comparing the SARS and the novel coronavirus. So this is the novel coronavirus and this is the original SARS. What's important here is that here we have a little animation. Like I said, these are not stable. These actually move. So what happens is that this part comes up. And so when we look at those different changes, we're actually looking on this little portion that is hidden before this part goes up. So here's a view from the top of the same region. So here's the comparison between the two separate ACE2 and their receptor binding domain, and here's where they are connected. And so you can see that there's actually quite a small surface that is responsible for that binding. So in the receptor binding domain, we also have insertions and substitutions. So you can see here a whole region, right? So the white ones are the dissimilar ones. We can forget about this one that has a lot of missing amino acids there. But if you see here, there's a clear distinction in these regions in terms of a couple of insertions and a lot of substitutions. And one of the things that was proposed in this publication in Nature, where they compared the human SARS coronavirus 2, that same bat, RAG13 novel coronavirus, a sequence that is very similar, pangolin, and then some of the older human and bat coronaviruses from the SARS family, they saw that there are specific changes that are more consistent between the human SARS coronavirus 2 and the bat, RAG13, but also in the receptor binding domain, you can see, so this is the third one from the top, you can see that specifically here are some receptor binding domain ACE2 contact residues. And those are more similar between the human and the pangolin as opposed to the human and the bat. Nevertheless, the bat overall sequence is more similar to that SARS coronavirus 2. So another area that's important here is the fusion or the cleavage site, sorry. And the cleavage site is actually a number of residues. It's important because what happens is the top completely comes off and the S1 subunit trimer goes through a conformation and helps with the fusion of the viral and host. So this is you can see how it's connected to the you set to two. It's connected a few places. And so this is the top S1 subunit. And once it comes off, we are left with the bottom subunit. The bottom subunit also has some very important domains, functional domains, which include the fusion protein, the cleavage site, and then the heptad repeats. And once that top comes off, they form the actual spike that helps with the fusion between the viral and the host cell membrane. So there also, there are specific changes. As you can see, that's where we see some of those important insertions. And some of those insertions, they keep the glycosylation residues that are responsible for how the glycans are attached to the whole protein from the outside. But nevertheless, this polybasic cleavage site is consistent. You can see here that it is inserted. And so it's important to understand potentially what might be the reason and what might be the functional implications of some of these inserts in these sites. Some of the studies that were done on the novel coronavirus, the original SARS coronavirus compared to MERS, is that this specific cleavage site predicted in the spike protein potentially related to viral transmissibility, right? So one of the potential explanations for that insertion is that it is important for cell-to-cell fusion. And then there are also changes at the substitution level between the heptad repeat region 1 in the SARS virus and the novel coronavirus. And so you can see not as many, but it's a fairly short sequence. So those are also, some of them are going to be more significant changes than others based on the type of change that we see. So looking in general, the conclusion from this type of analysis is, again, multiple layers of information that we have to connect together to be able to interpret what is really going on. So what we know from this analysis is that, of course, selection of the sequences could be starting from the public domain where we have a lot of data available. We could map the mutations at the nucleotide level and organize them by frequency of mutation. And the frequency of mutation changes reveal that the majority of the changes happen in very specific domains. And those specific domains are important for us to start and interpret how can we understand the differences in the function of this specific protein or others, as we saw before. And you can see that actually a lot of those significant mutations are around those critical regions, both in the spike glycoprotein on the receptor binding domain, as well as the S2 subunit, where you can see the heptad repeats and the fusion protein. And those are significantly different between the different groups of sequences. I think that it's important to link this type of analysis together, and so that's why, again, I'll just briefly go to this kind of visualization, because ultimately, when we look at a specific change or accumulation of mutations, here we can see that we have the nucleotide sequence, the amino acid sequence, and the reference aligned. Manually going through this information in FASTA files is quite challenging, trying to find the different regions in a protein and make sure that we are accurately finding the same sequence in the right domain. And also linking that to the structural changes, as well as understanding the evolutionary relationship between the different sequences. So we hope that organizing this information in such a way that we can easily link the evolutionary analysis with the sequence changes and with the structural information that we have on these proteins can help us make better sense of what is really going on. And so the output from the pipeline analysis is something that could be easily imported into this kind of view to make it easy to navigate and make sense of these changes. So as this is just a brief demonstration of some of the research projects that are being done with SARS-CoV-19 bioinformatics tools. I want to thank the Tauber Bioinformatics Research Center and the team that was working on some of the analysis that I showed, as well as some of the people that work with the LBRN and the general educational materials that we provide. I do want to mention that we will start a course with 4 LBRN members on COVID-19, but also some of the other viral data that could be found in public domain. And we are starting an online course on May 4th, but also there's going to be a specific LBRN announcement coming about this program on bioinformatics for infectious diseases. So that's all that I wanted to share today. Gus, maybe I could, we have a few questions here, or?
It sounds very good, Leo. Very nice. Very important tools, a very important analysis. So maybe we can entertain a few questions. If anybody has any question, can you actually either unmute yourselves or chat, pose the question in the chat back so we can see it.
So in chat, there's a couple of questions. One from Weishan Huang. To compare to amino acid sequences, is there a tool for making similarity plots? Alignment is great, but when there are many sequences, a similarity plot would help to narrow down and pinpoint the domains with variation. Yeah, so I agree, that's an important point. Maybe that's just a general comment. And another question is, do we have an idea of the structure or topology of these RNA domains? So I think some of those are known. Some of those are known in greater detail than others. But specifically for the spike glycoprotein and for the RNA polymerase, we do have the structures on PDB.
And interesting enough, Elia, the OLF8 variation, at least has been shown to may be associated with an evolving virus that may have a deletion in OLF8, and SP3 is part of the RNA pol that may also be relating to how proofreading capacity the virus may have. So it's kind of interesting, all of these mutations beyond, of course, the spike. We do know that the spike binds, the S1 binds to the ACE2 receptor about 20 times stronger. So that binding is probably contributing to higher level of infectivity. But we do know, and we have reported, that the new SARS spike is extremely much more fusogenic than the old SARS-1. So.
And the polymerase, yes, it has a specific region where those mutations seem to be accumulating, and so that might be related to the replication fidelity.
Yeah, the field is moving very rapidly. The UT Galveston reported recently the creation of an infectious cDNA clone of the entire virus, which means now they could be mutated, so specific mutations can be engineered within the virus to be able to address the role of these mutations in viral replication and maybe infectivity. So the field is moving very rapidly.
Exactly, yeah. So there's a question here. There are some spike protein 3D structures available by homologous modeling. How reliable are these for coronavirus 2 drug design projects? I think that's a good question. I know that there are a few groups making those predictions available. I don't know the quality of those predictions. I know there's one from University of Michigan, and then I think you can group from Google put out some of those. So I don't know if there are some experts in prediction that can answer that question.
Yeah, I think Sitirama left early before you guys another engagement. But prediction is prediction. If you have a really good cryo-M or electron, structure, then you could inform that prediction much better. So it should be much more accurate than it would be on a de novo prediction. So I would say if people know what they're doing, it should be fairly close.
Yeah, because typically those predictions leverage some of the known.
Exactly. Anybody else on your chat? Okay, I don't see anybody else here, either orally or on the chat. Do you see anybody else?
It says, how are you using this information for current vaccine development? Maybe that's to you, Gus.
Yeah, well, I think Eli mentioned the fact that the uh, spike glycoprotein is quite different. In fact, there are differences in glycosylation sites that may be shielding or altering the conformation of the glycoprotein. So the bigger question is, where are the B and T cell epitopes in these proteins, and how close they are to those glycosylation sites, and what the effect may be in immunogenicity. I suspect there is more of importance in the context of the entire virus, that individual proteins, when expressed without the rest of the proteins, may be conformed differently, in which case then there may be a better immune response than it would be in the context of the virus itself. So I think in this case, a live attenuated virus may not work, but heterologous expressed proteins may work, especially if they conform differently to expose B and T cell epitopes. We will know this answer hopefully within the next few months, because we have vaccine vectors that will be expressing the spike and hopefully a couple other proteins.
Okay, well, Claire said cool beans. So another question that there is, Wei Shan went through the website and did not see a tool for similarity plot of amino acid sequences. Can you please help locate it? Yeah, so maybe I'll just put in my email and we can set up a time
right.
To speak about that. And then there's another question. The Trump administration is proving whether the COVID-19 virus was lab made. What is the basis for this claim? So I think a good reference publication that is interesting to discuss in that context, we had a little insert here from that publication, and I think it offers some clues as to where you would look for those key regions. So this is a paper that basically offered three ways to look at that question of origin. So one is, how similar is that new coronavirus to other coronaviruses from different hosts? So the claim that the Chinese are making is that it came from bats. What we do know from comparing those sequences is that the bat-rat G13 sequence indeed is very close, but it's not in specific domains the closest one, right? So here we see the pangolin. That's where the theory of an intermediate host makes a lot of sense, that you might have some variation in one host, other variation in another host, potentially there's recombination in a host where both of those are present, right? And so the wet market theory looks feasible from just analysis of the data that was published there. Another question is the system for production of these viruses. Let's say they were engineered. If they were engineered to cause a specific harm, or if they were engineered to study specific aspects of those viruses, you would typically use a known system to test that and to conduct experiments. And so the pangolin is not a well-studied strain of coronaviruses, so I think that's an insight that they provided there. Their conclusion was it was probably not specifically engineered. But specifically engineered, with harm or just to study it, right? Those are two different things. And so I think the general idea that this might be harmful doesn't mean that it was necessarily, created to cause harm to humans. For different studies, it's a very common practice to try and implement, different mutations and see what's the result, and how that could escape from a lab. Probably possible, but that's the insight from the data. I think is those claims could be shed light on from analyzing sequences, understanding the differences and the similarities, and kind of looking at how those fit in with the overall idea of conducting experiments, looking at differences in those sequences, etc.
Yeah, so and I would add to that, Elia, that obviously, because there is reverse engineering of this virus, so there's an infectious cDNA clone of multiple human coronaviruses, those then could be modified at will at any point to address different issues. So is it possible that any of these viruses can be engineered? The answer is yes. There was a moratorium both in China and in the US of actually creating any viruses with gain of function capacity. And that was back in 2015. And, hopefully I'm sure the Wuhan Institute also was obeying, that order of not creating viruses that had gain of function. So for instance, the increased RBD binding is a gain of function. So I don't think anybody in the right mind will create a virus that would be more binding to an ACE2 receptor unless there was some kind of other purpose for that. So it does increasingly indicate that it may be an evolutionary adaptation of a specific virus. But don't forget that there are more than 500 different coronaviruses in bats. So and of course the Wuhan Institute and Galveston and others we're looking at all these bat viruses and how they differ and how they adapt. So is it possible then all those bat viruses, somehow, got free and infected some other animal? But I think the most plausible explanation is the pangolin is the some type of intermediate host that it came from the market or it came from some other event not necessarily at Wuhan, but somewhere else in China. There's also a lot of anecdotal information, the fact that these viruses, at least some of these viruses, may be circulating much earlier, maybe 6 months earlier. So the question is, if you have to go back and see how many different people died from pneumonia, whether any of those had this virus and maybe it evolved somewhere else and really expressed itself first time in China as opposed to coming from China. So there's still unanswered questions that can only be answered by looking over time where those viruses were and how they evolved.
Yeah, and another question here. Are RNA structured topology tools linked to the sequence analysis tools you presented? What can we learn by consideration of RNA structured topology in addition to its sequence and mapping to protein structure? So I think, Tom, you're the expert on the topology part of it. I would say that those are definitely linked in the sense that we have a lot of data from sequencing. We don't have a lot of different structures solved from different isolates. So we have a reference structure, and then we have a lot of sequencing data. So we can only start inferring what implications that might have, and then eventually test those very specific examples when we do know the topology. But I think that the connection is definitely very important.
Another area, Elia, that I think, and I've reviewed a bunch of papers recently, people are looking also at regulatory regions, leader sequences, subgenomic messages, because we do not know whether a number of messages created are equal among virulent and maybe avirulent strains, whether the pseudonut for frame shifting of ribosomes is affected in certain variants and so on. So in addition to the structure and function of proteins, it's possible that transcriptional regulation and replication characteristics of these viruses are maybe different, and they may be evolving differently between different strains that appeared in Europe versus the US versus somewhere else.
Yeah, and what we do know in terms of so different subgenomic regions, of course, are very important, and they play a role in regulation. I think there's a lot of unknowns there, but also with the idea of topology, one of the tools that I was showing in the beginning is this Q-align algorithm that actually looks for a more flexible comparison of structures that maintains some of the regional chemical properties by superimposition. So not superimposition from a molecular dynamics perspective, when you assume that the structure is very rigid, or at least you know exactly how it conforms, but when you're looking at global similarities, but at the structural physicochemical level. So some of those tools I think are very insightful with the data that we have, because there's a lot of variation that we're trying to still compare the impact of that variation on the structure. And so the other question here is, are the tools for knot prediction reliable? So that I actually do not know.
What was the question?
Are the tools for knot prediction reliable?
I don't understand the question. For knot prediction, what does it mean?
Like when you tie a rope in a knot, but I guess this is just, pseudo knots.
Yeah,
I don't know.
I don't know either.
RNA structure prediction. So yeah, so we talked about some of those predicted structures where different groups have put out some structures for either already solved cryo-M protein structures, or for some of the lesser known proteins. So those, as we talked about, they typically reference some known structure that is already there, and then they add on some of the variation. So how reliable, I think we don't know, but maybe next time we should do a panel of experts on structural biology and prediction.
Yeah. I think we're already almost 10 minutes after the hour, so maybe we could wrap it up at this point. Is there any other question that you want to answer, Elia?
I think that's all the questions. I think we did all of them. So
Okay,
again,
okay, great. Well, thank you very much, Elia. It was very informative. Everybody look forward to seeing some of this. We're going to be, obviously this lecture is taped, and we'll be going to be launching some of these tools throughout the LBRN network. Thank you for attending.